My God. Okay. Okay, the first question answer will be what? Just now I have explained you tRNA. Correct, na? So what will be the second question answer? Everyone said that. What? C answer. How it is C? How the second question answer will be C? I am not getting. How second question answer will be C? Seema, Varalakshmi, what you people has written? I am not getting. How it is C? How many times you people studied? Such a simple question it is. Look into this. Other than the DNA polymerase, how many times we studied? It is there in the first slide of the enzymes. Topoisomerase enzyme is required for removing the coiling. Helicase is the enzyme which open the double-stranded DNA. Then you're not understanding a point. Suppose this is single strand, this is another single strand of the DNA, which are acting as a templates. And you all people know very well, if the DNA polymerase enzyme wants to synthesize the nucleotide, first there should be an addition of the primer. Yes or no? Without adding the primer, how does the DNA polymerase is going to synthesize the new DNA? Who will add the primer? So that primer added by the RNA primase enzyme only. See, you all people remember, right? We never add the DNA primer because there is no DNA primer only. There is an enzyme called RNA primase. That is the one which will come and add the primer. I have explained it to all of you very clearly. What is the length of the primer? Also, I told you 11 plus or minus 1 is the length of the primer in our body. In vitro, within the laboratory, the primer length is 60 nucleotides. So friends, obviously we use the topoisomerase, we use the helicase, we use the RNA primase. Hence the correct answer will be the D. It's not C. Don't make simple mistakes, okay? Next, the enzyme used for joining the two DNA fragments is called as. Obviously, you all people know very well. What do we call it as? Ligase. Every student remember my statements very correctly. Listen, everyone. Most of the students will think that ligase enzyme is only involved in the replication. That is wrong statement. Every student listen. Ligase is involved in the transcription. Ligase is involved in the RNA processing. Ligase is involved in the repair. Ligase is involved in the recombination. Ligase is the enzyme which is involved in the multiple processes. In a simple words, what's the function of a ligase? The function of ligase is what? Sealing the nick. Everyone is saying that molecular glue. So what exactly it will do? It is the one which is actually involved in the phosphodiester bond formation. Yes or no, friends? Obviously, that is only our ultimate goal, right? We have to form the phosphodiester bond. Simple point it is. Now, nucleotide arrangement in the DNA can be seen by every student. What will be the answer for the question number 4? Naj. Okay. Now, my question to all of you is, every student answer my question, every one of you. This X-ray crystallography structure, I mean to say, the DNA was observed by the X-ray crystallography by two scientists. What are the name of the two scientists who observed the DNA structure by using X-ray crystallography? I have told to all of you in the molecular biology lesson when we are discussing it. No, Jayalakshmi, how it is Watson and Crick? Watson and Crick proposed the structure. Who observed DNA with the help of it? Very good, very good, very good. Rosalind Wilkins. Don't forget. Rosalind Wilkins and James Franklin. Is it or not? Rosalind Wilkins and James Franklin are the two scientists who observed the X-ray crystallography DNA by the X-ray crystallographic structure. Later, by using that, the Watson and Crick proposed the double helical. Every student, the Wakajaki fragments in a DNA chain with DNA chain growth. Everyone, option A says that, friends, polymerizes in the 3' to 5' direction and forms a replication fork. Every student know it. I have told to all of you many times. Friends, whether it is a leading strand, whether it is a lagging strand, doesn't matter, friends. Both the strands are going to synthesize in the 5' to 3' direction only. Why others are not answering? Once again, remember, whether it's a leading strand or lagging strand, both are synthesized in the 5' to 3' only. All the students listen. So here what is happening? One strand is moving towards the replication fork. One strand is moving away from the replication fork. But what is the common point? The common point is that both are going to be synthesized in the 5' to 3' direction only. There is no doubt. So it proves semi-conservative nature of the DNA replication. No, I'm telling you, stop your videos. Please don't play your videos and do not disturb me in the middle of the class. Okay. Next, option C, polymerize in the 5' to 3' direction and explain 3' to 5' DNA replication. What do you mean by that? Why the option C will be the correct option? We all people studied. Our parental DNA is in the 3' to 5'. Every student try to understand me, friends. I have told to all of you multiple times. Our DNA polymerase enzyme, everyone remember, DNA polymerase enzyme read the parent strand or the template in the 3' to 5' direction only. Then obviously the daughter strand has to be synthesized in the 5' to 3' direction only. Hence the correct one will be what? Option C. Question number 6. Reverse transcriptase is the question which is asked in your set exam also. What will be the answer? And most of the students are getting confused between a DNA dependent, RNA dependent. Why you are calling DNA dependent and why you are calling RNA dependent? All the students listen. Every student listen my words. Every one of you very carefully. Everyone listen. See friends, there is a DNA. Everyone remember. Friends, in the computer, we are having a hard disk. What is the speciality of the hard disk? Hard disk stores the entire information. My friends, similarly, DNA is the one which stores the information. DNA is the one which gives rise to the RNA. With the help of DNA only, RNA can give rise to the proteins. So everything is under the control of DNA. So when I am synthesizing the RNA from the DNA, which polymerase is involved? RNA polymerase is involved. But the point is what, friends? The signal is coming from what? From where? The guidance is coming from where? The guidance the guidance is coming from DNA. DNA dependent RNA polymerase is the one which synthesizes RNA. But that is not the question asked. Reverse transcriptase. We all people know that 90% of all the organisms are having DNA as their genetic material. Except viruses. Viruses are having RNA as their genetic material. So friends, in case of a viruses, RNA stores the information. Now, what is reverse transcription? From the RNA, we are synthesizing the DNA, which means that the guidance also should come from the RNA only. But we are synthesizing DNA, right? So what enzyme we require? We require DNA polymerase, which is dependent on the RNA. So that is the reason the enzyme involved is RNA dependent DNA polymerase enzyme. And absolutely the answer will be the D. Every one of you look into the question number 7. Telomerase is an enzyme which is, I have told to all of you in the eukaryotic DNA replication very, very clearly, and I have shown you the animation also. What did I told you? B. How Varalakshmi it is B? Huh? Everyone is not answering the questions very correctly. Who said that it is a repetitive DNA? I'm not getting. Who said that it is a repetitive DNA? At least one fellow. Very good, Kirtana. Very, very good. Good. Good option. Good. See, Varalakshmi, look into this. This is the eukaryotic DNA. Correct, na? The ends of the eukaryotic DNA, we are having the telomere. Correct, na? What is the function of a telomere? I have shown you in the slides very clearly, right? Telomere protect the DNA from the degradation. Telomere extends. What it will extend? It will extend the length of the DNA. Friends, please don't get confused with this particular point. And see, Varalakshmi, what you thought is, you thought telomere. Telomere is the repetitive DNA. They did not ask the telomere, right? They asked an enzyme. The enzyme is the telomerase. The telomerase enzyme, I have shown you in the animation also, no? The telomerase is the enzyme which is formed by the RNA plus protein. Every student follow me correctly. It is formed by the RNA plus protein. Since both RNA and protein both are involved, friends, that's why we are calling it as the ribonucleoprotein. You're not reading, people. Okay. Question number 8. DNA template sequence of a CTGA TAGC is transcribed over mRNA is. Everyone. What will be the answer for question number 8? Question number 8. Good. Question number 8. Very good. So obviously what it is, the answer will be the B. You know, right? During the process of transcription from the DNA template, during the transcription from the DNA template, complementary mRNA is formed and thymine is replaced with what? Thymine is mostly replaced with the uracil. Please do not get confused with this particular point. In the complementary strand, don't get confused. It's a very direct and straightforward question. Good. Then question number 9. During the replication of a bacterial chromosome, DNA synthesis starts from a replication origin site and it's very simple. I have explained you 4 to 5 times. Everyone, question number 9. Wonderful. Friends, all the students remember whether it is a prokaryotic organism or whether it is a eukaryotic organism. What I mean to say, whether it is the circular DNA, whether it is the linear DNA, all the students keep this point in your mind. The type of replication will be the bidirectional replication only. Okay. The type of replication will be what? Bidirectional replication only. Kindly don't get confused with this particular point. Never ever confuse with this. Okay. So now answer should be D. Very simple point. Very good. Question number 10. The enzyme which helps to cut one strand of the DNA complex to release tension of coiling of 2 strands. I have explained it to all of you many number of times. Whichever is removing the coiling or whichever is removing the tension, you can call it topoisomerase, but widely called, my friends, gyrase. Ligase will seal the nick, polymerase will do the replication, helicase will open the DNA. But the question asked is which is removing the coiling, which is removing the tension, which is undoubtedly topoisomerase. Histones. Every student has to answer the question. What are histones? Question number 11. Question number 11. Question number 11. Obviously, how many number of times we studied recently also, right? Histones are the basic proteins. Very, very good. Every student quickly answer my question. What are the name of the 2 major amino acids that are present on the histone tails? What are the 2 major amino acids that are present on the histone tails? Everyone, Ramana, Narayani, everyone, quick. And Kirtana, what is the other amino acid? Don't forget. Very good. So it's a basic protein, right? It's not asparagine. It's not asparagine. It's arginine, lysine and arginine. Botany students has to read the amino acids compulsory. Okay. In zoology JL syllabus, amino acids also there, but your syllabus it is not there. But it's very important, friends. One gene, one enzyme relationship was established first time in. It is a direct question, friends, but I want you people to understand. Anyone given the answer for question number 12? Question number 12. Anyone? Very, very good. Very good. Very good. This is the direct name of the organism only. You have to remember, which is the Neurospora crassa. I want every one of you to remember this compulsory, friends. That's the direct name of the organism. Next, question number 13. The transformation principle of pneumococcus as found out by Avery, McLeod, and McCarthy was, you know, right? In your set exam, the same 3 scientists only asked in the set exam in a matching question. I don't know whether you recall it or not. What's the question number? Answer for the question number 13? Obviously, what? DNA. Good. How many pair of nucleotides are present in one helix of a bDNA? My friends, this you people can answer when you study the nucleic acid biochemistry. If you don't study the nucleic acid biochemistry, you know, you may guess it, but you have to guess it correctly. All the students listen. Okay. Every one of you answer my question. In our body, I'm asking a question to all of you. In our body, what kind of DNA is present? Is it a bDNA or is it a cDNA or is it a zDNA? Everyone. In human body, what kind of DNA is present? bDNA, cDNA or zDNA? Every student answer, what is the type of the DNA that is discovered by the Watson and Crick? The Watson and Crick also discovered the bDNA only. My friends, every student listen. This is called one helix. It is called how many helix? One helix. One helix contains 10 base pair. But my friends, if you look into the cDNA, it contains 10.5. If you look into the zDNA, it is containing 11.5 to 12. So I want you to remember for bDNA also, cDNA also, as well as the zDNA also. Hence the correct answer will be A. So look into the statements very correctly, friends. Read the following 4 statements. In transcription, adenos, what is the question asked? The question asked, the above statements are correct. In transcription, adenosine, adenine can be replaced by the uracil. Is this statement is a true statement or a false statement? Everyone say yes or no. The statement 1 is a correct statement or a wrong statement. It is obviously a false statement. Regulation of lac operon by repressor is referred as positive gene regulation. Every student has to answer my question. Option 2 is a right statement or a wrong statement. Everyone say yes or no to me. Second statement is a wrong, true statement. It is a false statement. So I want every student has to answer my question. Then what is the name of the positive regulatory mechanism? I hope you all remember. Positive regulatory mechanism by CAP gene. Correct na? Positive regulatory mechanism by what? CAP, cyclic AMP. Wonderful, wonderful. Then option 3 says that the human genome has approximately 50,000 genes. Is that correct? No, it is having 30,000. So it is also wrong. Hemophilia is a sex-linked recessive disease. Is it a correct statement or a wrong statement? Every one of you. Hemophilia is a sex-linked recessive. Obviously true. Hence, my friends, the answer is D. There is only one statement is the correct statement. Wonderful, wonderful, wonderful. Okay. Next, 16th question. In a negative operon means you all people know that. Negative means what, my friends? There will be a no cap protein is involved. So option D will be the wrong. Co-repressor binds with the repressor. Co-repressor does not bind with the repressor. Co-repressor bind with the inducer. Cyclic AMP have a negative effect on the lac operon. Every student, what will be the answer for the question number 16? Anyone? It cannot be C. It cannot be B. It is A. Okay. My friends, here you know what is co-repressor. All the students has to understand my questions very correctly. Without co-repressor, all the students remember, without co-repressor, there is a no possibility that the repressor never ever be active. If the repressor wants to be active, friends, there is a requirement of co-repressor. Then co-repressor, when it associated with the repressor, then it will go and block the operator. You're getting my point. That is only we can call negative gene regulation. Then option A will be the correct. Here it is saying does not bind. Co-repressor never bind with inducer. That is a wrong statement. So correct answer will be the A. No need to confuse. It's easy. Question number 17 says that transfer of genetic material from one bacterium to another bacterium in the transduction process is through every student, which is bacteriophage. You all people commonly know about it. Direct question. Telomere and eukaryotic chromosome possess a short segments of. Friends, it's a very important, friends. Telomere repeats what? It is a direct question, NCERT question. Telomeres are the rich repeats of the guanine. All the students always remember this particular point. Please do not forget that. Okay, you all people know that. We have seen the diagram multiple times. Every student answer the question. Refer the given figure of nucleosome and select the ABC. You only tell me, friends. So tell me, everyone. So what will be the A, friends? Good. So histone, DNA, and H1 histone. You all people know that H1 histone is called as the linker protein. Correct na? And this thread-like structures, or what do we call it? That is only DNA. And this round structures, what do we call it? They are the histones. Very, very simple. Hence the correct answer will be C. Very good, very good. Question number 20. The process of transformation is not affected by which of the following enzymes? Very, very good question it is. My friends, listen the question. The question says that which is not affected by which of the following enzyme? Very simple, straightforward question only, na? How it is D, my friends? It's a really good question. Think it. Anyone else? What will be the answer for the question number? What is the question number? Question number 20. So, yeah. Everyone. See, friends, remember, RNAs, good. RNAs is responsible for what, friends? RNA is responsible for RNA digestion. Lipase is the one which will cut the what bond? Ester bond. And peptidase is the one which will cut what? Peptide bond. So, but in case of transformation, it is only with respect to the DNA, right? Only DNA will be isolated from one cell and we will introduce it into the another cell. So then there is no role of RNAs, lipase, peptidase. Hence the correct answer will be the C. Very good. Next, question number 21. Select the incorrect statement regarding the DNA replication. What is the question? Option A says, leading strand is formed in 5' to 3'. Wonderful, right? Okazaki fragments also 5' to 3'. There is no doubt about it. DNA polymerase enzyme always synthesize the daughter strand in the 5' to 3'. Absolutely true statement. And option D says that DNA polymerase catalyze the polymerization in 3' to 5', which is absolutely wrong statement. That cannot be the answer. Hence the correct answer will be what, friends? D. It's not difficult questions at all. Okay, now look into the question. This is a very good question. Question number 22. Identify the labels A, B, C, D in a given structure of a tRNA and select the correct option. My friends, I am explaining for all of you once again. Wherever all the students has to listen, wherever you can see the 3' hydroxyl group, that will become what? Amino acid binding site. Every student has to remember for the exam point, every student at the amino acid binding site, that is at the 3' hydroxyl. I have shown you the picture. I have explained you very briefly. It contains a sequence called ACC. This is called as what? DHU loop. My friends, please don't confuse. What did I told to all of you? Friends, this particular D loop will help here. Correct, na? And this is called as what loop, friends? This is called anticodon loop. What did I told you? No, this will be the T loop. And don't, sorry. 1 second. This is correct. This is the amino acid binding site. This is the DHU loop. This will be the anticodon loop. And what about this? This will be the DHU. Don't get confused. I have very clearly told you in the exam. Every student listen. This is a 3' hydroxyl group. One loop will help this to recognize the correct amino acid. And this is what loop? Anticodon loop. Anticodon loop has to get a positive signal, right? So that can be, that particular signal will comes from this particular T loop. Very simple. One is guiding the amino acid and another one is guiding the anticodon loop. Hence the correct answer will be the B. It is also straightforward, direct question. Next, you all people know that. What is this? Question number 23, everyone. In one polynucleotide strand of a DNA molecule, the ratio of A plus T by G plus C equal to 0.3. What is the A plus G by T plus C ratio of the entire DNA molecule? Everyone. What will be the answer? You can always calculate like this, right? A plus G by T plus C. Everyone. What will be the answer for this? See friends, you should all, what is the answer for the question number 23? What is the answer? You're not understanding the question. The question says that how it is A, friends. What is the answer for question number 23? I want you all people remember one common statement. Every one of you remember one common statement. Okay. Whomever did not understand, everyone remember. According to the Chargaff's rule, purines should be equal to the pyrimidines. Correct, na? Purines should be always equal to the pyrimidines. What they have given here, A plus by T by G plus C is 0.3 in one strand. A plus by T, G plus C in one. See, this is one strand. One strand. Please mute your videos. I don't know why you people don't do that. Mute your videos. Please do not disturb me in the middle. Okay. Now, every one of you listen. Listen, everyone, whomever did not understand. Here, A plus T by G plus C ratio in one strand, it will be the 0.3. This is a fixed. What is the question they ask? What is the A plus G by T plus ratio of the entire DNA molecule? And all the students remember, purines equal to pyrimidines. You should always equal to the 1. If this will be 0, no need to confuse here. If it will be the 0.3, what will be the other strand? Other strand will be 0.7. Then it will be total equal to how much? Total equal to 1. Most of the students will choose the answer as the B as the 0.6. That is not the question they asked. They asked one strand having the sequences, that ratio is 0.3. They did not ask what is the ratio of the second strand. They did not ask that. They asked what is the total A plus G by T plus ratio of the entire DNA molecule. If one strand will be 0.3, you should always equate it to the 1. Then other strand should be 0.7, which will be how much? Which will be equal to the 1. Do not get confused with that particular point. So that's why you always remember A plus G by T plus C should be equal to 1. So they have given already 0.3 in one strand, other strand should be 0.7, then total should be 1. Tell me anyone did not understand. I will re-explain question number 23. Is it clear or not? But friends, in a given question, instead of asking the entire DNA molecule, if they asked for second strand, then the answer should be 0.7. Then the answer should be 0.7. They did not ask for the second strand. They asked for the entire DNA. Then obviously it should be 1. No need to confuse. Okay. What is there in this? This question is very easy, friends. According to the Chargaff's rule, every student remember this formula enough. A plus G by T plus C always should be 1. Right. What they have given on one strand of the DNA, A plus by T by G plus C equal to 0.3. This is the ratio. Please don't consider as the percentage. If it is on one strand, it is the 0.3. Now, they never asked for the other strand ratio. They asked what is the total DNA ratio. That is the question they asked. What will be the total DNA ratio? It should be equal to 1, right? So obviously the answer should be the 1. Understand clearly or not? That's a very simple question. Wonderful, wonderful. Chemically, RNA is reactive and stable as compared to DNA. Every student, what will be the answer for the question number 24? You know, right? RNA, everyone, what will be the question number 24? RNA is the more reactive and which is the less stable. Friends, all the time remember DNA is a very, 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 very highly stable molecule. Never ever forget that particular point. So the same diagram I have explained you when I am explaining you the molecular biology, the same diagram I have explained you. Tell me everyone, given diagram represents the components. Why we have to look into the options first of all? Without looking at the options only, you people only can give me the answers now. We no need to look into the options also. Okay. Tell me every student, what will be the A? What will be the A? What will be the A? Friends, A will be the promoter. Very good, very good. What will be the B, friends? What will be the B? Good. Terminator. Very good. So what will be the C and D? What will be the C? What will be the C? Very good. C will be the template. Wonderful. Okay. Next. What will be the D? What will be the D? Obviously, a coding strand. Correct na? So obviously answer will be C. See friends, every student has to remember the statement. I have explained it to very clearly. Friends, whichever is the template, it is always non-coding strand. Whichever is the non-template, I told you that will become a coding strand. Yes or no? Template will be the non-coding and non-template will be the coding. I have explained you very clearly in the transcription. Okay. Question number 26. In transcription in eukaryotes, heterogeneous nuclear hnRNA is transcribed by. Every one of you very quickly. Which is what? RNA polymerase 2. Friends, always remember friends. RNA polymerase 1 can synthesize rRNA. RNA polymerase 2 can synthesize mRNA. RNA polymerase 3 can synthesize tRNA and all other small type of the RNAs. Very good, very good, very good. Perfect. Okay. Question number 27. Very good question, right? Every student has to answer the question correctly. What would happen if a gene encoding a polypeptide having 50 amino acid, 25th codon is the UAU, it is mutated with UAA. What happens? Simple question. Every student has to answer the question. 27. Okay. See friends, you only try to understand. Everyone remember, there are 50 amino acids are there. What happened to the 25th codon? It is converted from UAA to UAA. And we all people know very well what is UAA. No. Someone said B. No option. Look into this. UAU converted into UAA. UAA is what, friends? UAA is the stop codon. So the ribosome never ever moved to the stop codon. So what is happening now? Only the 24 amino acids are getting translated into the protein. From the 25th amino acid, everything will be stopped immediately. Correct na? Hence the correct answer will be what? The correct answer will be the option A. Question number 28. The regulation of gene expression occurs at the level of. Wonderful. I want every student has to answer the question. If you miss the question, I don't encourage anyone. I want you to answer this question correctly. Question number 28. I have explained you this specific point when I'm teaching to all of you. Wonderful, Morally. Very, very good. You all people are not reading correctly, not analyzing the question correctly. All the students remember, every one of you remember my words. Friends, when it comes to the replication, replication is not the point where the gene expression will start. Whomever given the wrong answer, remember. Friends, remember. Transcription is the point where the gene expression will start. Okay. Now let me ask you one small question to all of you. Among the A, B, C, where the maximum gene transcription occur, every one of you answer. Where is the maximum gene transcription takes place? Is it a transcription or a splicing or a translation? Everyone. Transcription. Seema, you forgot everything. Huh? Just now I told you, right? The gene transcription starts. Every student listen. Every student listen. The gene transcription starts. The gene expression starts at the transcription. The gene expression continued during the splicing. The gene transcription will be maximum at the translation. Friends, everyone think with common sense. What is the ultimate goal of a gene? To synthesize the protein. So where I am synthesizing the protein? I am synthesizing the protein at the translation, which means that maximum gene is getting expressed in the translation. Then obviously 28 will be C. So maximum gene expression occurs at the translation, but gene expression occurs at the transcription also, processing also, translation also. Hence the correct answer is D. Question number 28, answer will be D. Friends, don't confuse, please. Find out the wrong sentence about heterochromatin. If anyone makes mistake, I never agree. How many times I have explained it to all of you? Same statements only I have explained to you. It never be transcriptionally active. It is very tightly packed. It stains dark. It's late replicating because it is tightly coiled. How does the replication transcription will take place? Wonderful, wonderful. Question number 30. Other than DNA polymerase, which are the enzymes involved in the DNA synthesis? Of course, all. Okay, this question is repeated. Okay, what does the lac refer to in what we call the lac operon? So lac represents what? Lactose. Correct, na? Lac represents what? Lactose. It's a very good question, friends. Everyone quickly answer the question number 32. The error rate of changing an incorrect base with another incorrect base during the proofreading is? My friends, I am telling to all of you, very good, very, very good. Here they have given 1 in 10,000 base. My friends, even though they give 1 into 10 power 6, that is 10 lakhs also correct. Even it is 10 power 6 also correct. But you know, recently we understood that DNA polymerase enzyme is making only one mistake out of 10 power 7 nucleotides added. What do you mean by that? Out of 1 crore of nucleotides added, out of 1 crore, it is making one mistake. So if they give 10 lakhs also, 1 crore also, 10,000 also, always choose the highest one. Always choose the highest one. And it's a direct question. DNA fingerprinting refers to. It is not there in your syllabus, but it is a very direct question. I mean to say in your JL syllabus, it is not there. Everyone commonly know, right? DNA fingerprinting. Who has given the answer as B, C? Huh? What? What is the answer for question number 33? Why this many answers? How analysis of DNA sample by using imprinting devices? No, DNA fingerprinting is not an imprinting device. How the answer should be B? Huh? DNA fingerprinting is molecular analysis of the profiles of the DNA samples. Correct now, friends? We can call it them as the VNTR. Always remember, why do we use the imprinting devices? DNA fingerprinting only a method. It is a technique we used to separate the DNA fragments. You know, right? In the crime scene, we will get the hair sample, we will get the semen sample, we will get the blood sample. So from that, what we will do? By using DNA fingerprinting only, we can analyze the DNA sequence. Correct. Answer will be the A. In negative operon, we already studied. I don't know why it got repeated. The question number will be the answer will be the A. 35. Genes that are involved in turning on or turning off the transcription of a set of structural genes are called as. Everyone, don't confuse with the question. How regulatory genes? Huh? Who has given? Anjum, how the regulatory gene will be the answer? The question says that genes that are involved in turning on or turning off the transcription of a set of structural genes, which is called operator. Friends, you remember the concept correctly or not? If RNA polymerase enzyme cross the operator, then only the structural genes are going to be active. If it is not crossing the operator, friends, there will be no way of activating the structural gene. Then obviously the answer will be the operator. Simple, right? 36. If the gene encoding the lac, I want every student has to answer the question number 36. I believe that you got the concept. if the gene encoding the lac repressor is mutated so that it can no longer bind to the operator, will the transcription of the operand occur or not? Think it, think it. It's a very, 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 very good question. Tell me, Naj, what is the answer? Anyone? I want specific answer. Don't give random answers. Wonderful, wonderful. See, friends, the question is crystal clear. If the gene encoding the lac repressor is mutated so that it can no longer bind to the operator, you tell me, friends, repressor got mutated, which means what? Every student try to understand. Repressor got mutated means operator is free. Friends, if the operator is free, RNA polymerase can easily cross the operator. If the RNA polymerase cross the operator, it can synthesize lac Z, lac A, lac Y, lac A continuously. And option C will be the correct answer. And they are not making the question difficult. If they ask a negative gene regulation, positive gene regulation, then you should consider option A. But they did not ask whether it's a negative, whether it's a positive. They just given one case, that is the repressor got mutated. Repressor mutated, operator is free, then everything occur continuously. Option C will be the correct. Transcriptional regulation in prokaryotes can occur by. I have explained it in the same statement. You have to think it. I don't encourage anyone if you give a wrong answer. Think it correctly. It's very logical question, friends. It's very, very logical question. Question number 37 is very logical. You know why this question is very logical? Every student listen. Every student listen. Any one fellow given the answer AC. Why others are not giving? Varalakshmi, Ramya said D. All the students listen. Here they are asking a question with respect to the transcription. They have considered the lac operon also. They have considered the tryptophan operon also. My friends, whether it is a lac operon or a tryptophan operon, we are synthesizing something. We are transcribing the genes. So they are talking about the transcriptional regulation. It is not only the process of gene transcription. They're talking about regulation. Repressor binding an operator and preventing the transcription. It occurs in the transcription, right? Of course, yes. An activator binding upstream from a promoter and positively affect the transcription, right? I have explained it to you multiple times. Friends, activators or enhancers are the same, which will enhance the transcription. Option C says that different promoter sequences binding RNA polymerase more tightly, resulting in more effective transcriptional. Of course, true. Because we studied, right, RNA polymerase will come and associated with the promoters only. That is also method of regulation. Hence, the correct answer is D. Why most of the students given a wrong answer? You are all only thinking about what happens in the transcription. No. That is not the question, friends. The question asked is transcriptional regulation. I hope you're following me correctly. How the process of regulation of transcription occur in the prokaryotes. What are the different methods? By using 3 methods, we can regulate it. It's a good logical question. Question number 38. Process used for the amplification of multiplication of DNA for the fingerprinting. You all studied from multiple times, which is the PCR. Right. Polymerase chain reaction. Question number 39. Direct question. Lactose operon produces what, friends? One is called beta galactosidase enzyme, which will break down the lactose into glucose and galactose. Permease, which bring the lactose inside. Transacetylase, I have told you very clearly. Friends, the functions of transacetylase is not clear so far, but it is meant for transfer of acetyl group. It is meant for stability of the operon. Hence, the correct answer is B. Satellite DNA. Anyone answer the question? What is the answer for the question number 40? Every one of you? Good. Satellite DNA normally does not code for any protein. I don't know whether you remember this or not. I have explained this in the transposons concept. Satellite DNA is a highly repetitive DNA in our body. Highly repetitive gene sequence. It never code for any protein. Then you can ask me, sir, if it is not going to code for the protein, what is the function? function is, friends, only regulation. Simple point. In prokaryotes, gene regulation occurs at the level of. Now look into this. Seema, now you look into the difference of this particular question. They're asking, now tell me, everyone, what is the answer for question number 41? Don't confuse. Don't confuse for the question number 41. Everyone, what is the answer for question number 41? Every one of you. Why others are not answering for question number 41? See, the question says that where the gene transcription regulation starts. They are not asking, friends, where it occurs maximum. They're asking where it starts. It starts at the transcription. Wonderful. Question number 42, the regulation of tryptophan synthesis in E. coli is an example of affecting a gene expression through. Every student. The same question asked in the set exam, the last question of the set, 100th question. That is the process of attenuation. Question number 42. What is the question? Yeah, of course, transcriptional control, because we are controlling the gene expression, right? 5 genes are there. Good. Question number 43. Transcription in prokaryotic cell is initiated at a promoter using one of the 3 RNA polymerases, RNA polymerase 2. Then, see, but the question is what? What is the question? Transcription in prokaryotes. This will be the wrong option, right? Because, my friends, in case of eukaryotes, there are 3 RNA polymerases are there. But in case of prokaryotes, there is only 1 RNA polymerase, right? This option will be wrong option. Correct, na? Option B says that initiated at a start codon with the help of initiation factor and the small subunit of the ribosome. This is also a true statement. I agree with all of you. But no. This cannot be a true statement, my friends. Okay. This cannot be a true statement. Option B cannot be a true statement because every student listen. They are saying that transcription starts at the start codon. That cannot be correct, right? Transcription starts at INR. That is called initiation region, not at the start codon. Option B is wrong option. Option C says that it initiated at a promoter and uses only 1 strand of DNA. Of course, that is the template. Undoubtedly, option C is the correct. Correct, na? Undoubtedly. Very good. It never terminated at the stop codon because you know, right, with the help of a row protein, it will do it. Stop codon will be in the eukaryotes. Option C is the right. DNA replication is and what process? This is also very, very good question. DNA replication is what? Energy consuming process. We keep on consuming endergonic and does require energy because it is taking energy, consuming energy. 45, which one of the following triplet codes is correctly matched with a specific for an amino acid in protein synthesis or start or stop codon? And you know, right, what will be the answer for this 45A? What is the answer for 45? So it should be what? UCG, no. UGU, leucine, no. UUU, no. UAC. UAC will be the tyrosine. Correct, na? UUU code for what? UUU code for phenylalanine. Correct, na, friends? UGU code for what? Cysteine. Is it or not? Then what about this particular UCG? UCG code for serine. The only correct one is UAC. Is it clear, everyone, all the 45 questions? Now, if you have any kind of a doubt, please let me know. I will share you the complete explanation to all of you. Every single question explanation. Clear, everyone? And once again, keep the point in your mind. Today is 17th. Next week, that is the 23rd, I want you to write the exam on genetics. Okay. Prepare the genetics exam. I want you to write the exam on this one. Next Sunday. Okay. I'll share you the explanations. That's perfect. I'll see you in the tomorrow morning 5:45 class. We see tomorrow. If any problem occur in the app, I will directly take to the Zoom itself. Okay. We will continue the ecology. Thank you. Thank you all. I'll see you in the next class. Thank you. Thank you.